Hello and welcome back to BIO4YOU. Okay, in this video we're going to talk about dihybrid cross for genetic inheritance chapter 4. Let's check it out. Okay, dihybrid cross, what does it mean? If monohybrid cross, we do a cross for one trait or one characteristics. For dihybrid cross means we're going to observe two characteristics. Okay, so in this example, we're going to observe the trait for seed color, iaitu ciri-ciri warna seed biji benih itu, and the other one is seed shape, bentuk seed tersebut. So it involves crossing between two individuals of true breeding, i.e. homozygous, with different phenotypes. Yellow round seed. Okay, so here there are two traits, yellow and round. Okay, homozygous dominant with the genotype Y besar, R kecil, R besar, R besar. So capital Y, capital Y, capital R, and green wrinkle seed. So green for the color, wrinkle for the shape. For this is also homozygous, but it is recessive. So you have the small y with small r. Okay, so again, allele Y. So allele is dominant allele for yellow seed. Allele Y with small letter is recessive allele for green seed. Allele capital R for dominant for round seed. Allele with small r is recessive allele for wrinkle seed. So for Mendel's experiment, still even for dihybrid, will do by three steps. Okay, produce the B generation, i.e. the true breeding, and then producing the F1 generation, and then produce the F2 generation. Mendel will study traits in the generations of pea plant. Okay, so for dihybrid cross, the production of P generation, i.e. the parental generation, the true breeding parents, where we have the yellow round seed with capital Y, capital R, and green wrinkle seed, small y, small r. Okay, so these are the actual diagram, actual photo of the green wrinkle seed, and this one is a yellow and round seed. Okay, inilah biji benih untuk pokok P ini, dan inilah biji benih untuk pokok P ini. Okay, producing the F1 generation, the true breeding varieties, okay, is cross fertilized. Cross fertilized means saling dikacutkan antara satu sama lain, and the results, okay, all of the F1 offspring have yellow and round seed. Okay, as we have predicted this because yellow is dominant trait and round shape is also a dominant trait over the green and wrinkle. Okay, so then producing the F2 generation, F1 is allowed to self-fertilize to produce F2 generation. Again, for F1 self-fertilization, we are using the same generation of F1 crossed with F1, i.e. they are siblings, okay, from the same generation, same batch of generation. Okay, so and the result for the F2 generation is a mixture of, we have the round and yellow, round and green, something new, wrinkle and yellow, also new, wrinkle green, just like the parents. Okay, so Mendel has concluded and comes into making his second law, Mendel's second law, i.e. law of independent assortment. So by the end of meiosis, genes on pairs of homologous chromosomes separate independently to another pairs of homologous chromosome. What does this mean? That, okay, previously when he studied monohybrid, i.e. just with one observing one trait, one characteristics, we agree that based on his first law that the gene will be separated, okay, and pass into their offsprings. Okay, law of segregation means they will be separated. Okay, for in this case, law of Mendel's second law, law of independent assortment. If we observe two traits, let's say the color of the seed and shape of the seed, that these two traits, they are also independently assorted and mixed with each other. It is not necessary that the dominant always stick with the dominant of the others. They can mix with they can produce a mixture of combination. Means dia bebas untuk assort, okay, untuk susun. Alright, we shall look on the next diagram, which I hope will give you a better and clearer idea about this. Okay, so we see from this diagram over here, we see this parent over here. Okay, at the end of during meiosis 1, you can see this what could happen, and then at the end of meiosis 2, these are all the possible combinations of the gametes produced by the parents. Gametes means either the sperm or the ovum combination. So let us look at possibility 1 on one side. So let's say for meiosis 1, both of the black, i.e. the dominant traits, okay, sets on one side, and the recessive trait set on the other side, okay, Y kecil, R kecil, R kecil. So you can ends up with this one cell, okay, after meiosis 1, it only carries Y kecil, R kecil, okay, and it ends up with the sperm is either having Y kecil, R kecil, and Y kecil, R kecil, and this is the genotype as what I've mentioned earlier. And the other cell, okay, after the cytokinesis of meiosis 1, Y besar, R besar, it ends up with the sperm with capital Y, capital R, capital Y, capital R. So as being written in this genotype here. Okay, on the other hand, there's also another possibility whereby after meiosis 1, through Mendel's second law, a law of independent assortment, means that the chromosome, they are independently to assort. Mana dia boleh menyusun dengan secara bebas je. Tak kisah tak semestinya yang merah mesti one side, black on the other side. Tak semestinya. Because during cell division, this chromosome arrangement is random. Random tu maksud dia rawa, okay, secara tidak ditetapkan. Okay, so what we see the possible of after meiosis 1 for this situation of possibility 2, one cell is having Y besar, R kecil, on the other hand, Y kecil, R besar, and it will produce the sperm, let's say, with Y besar, R kecil, Y besar, R kecil, Y kecil, R besar, Y kecil, R besar. So another different sets of possibilities over here. So again, for conclusion for this slide, that law of independent assortment. Okay. Alright, so this slide is showing this example of the genetic diagram on dihybrid cross and also including the phenotypic ratio of 9331 for the F2 generation. Okay, so what we're going to do here is the similar genetic diagram with the monohybrid cross, but with two traits. Ada dua ciri-ciri kat sini. Yellow for the color and shape is round. And this one, the color is green and the shape is wrinkled, mana berkedut-kedut. So this is the parent genotype. Okay, so the parent generation. So you must write P over here. If you didn't write P over here, your marks for this line will be gone. Okay, you will not be awarded mark for this line. So for the gamete, possible gamete, i.e. sperm, since this is a male parent. Okay, so the sperm could only carry either one of these, okay, Y. So since Y besar is the same, capital Y, so we're going only to write once Y besar over here, and for the R, since both are the same, we only write it once over here. Okay, so same goes with the other parent. This is the ovum with Y kecil, R kecil. Okay, and when you cross these two, you have Y besar, Y kecil, so Y with Y, R with R, R with R. Okay, so in terms of genotype ratio, all are capital Y, small letter Y, capital R, and small letter R. And in terms of the phenotype ratio, what we can see that all of the F1 offspring, F1 generation, is all are yellow and round seed. Okay. Alright, next step, when we do self-cross, Okay, so what I mean by self-cross is that we're crossing F1 with F1 generation. Okay, so yellow round with yellow round. So we will end up with slightly different over here, different possible of gametes. Okay, so there are four possible types of sperm over here and four possible types of ovum over here. Okay, so this one type of sperm could carry for the genetic for the color, it carries Y, capital Y, and for the shape it carries capital R. So Y besar, R besar. So the next possibility of the sperm formation is that it still carries the Y besar but carries R kecil on the other hand. Or you could also have Y kecil mixed with R besar and Y kecil mixed with R kecil. So these are all the possible four types of gametes. Same for the female, for the ovum. So for this one, we don't usually do the line crossing here. It's very tedious and very confusing. Hence, for dihybrid cross, for the F2 generation, we must use a Punnett Square to do this. Okay, so here is the Punnett Square. We have the sperm types here. Y besar, R besar, Y besar, R kecil, Y kecil, R besar, Y besar, R besar, and then it is crossed with same type of the ovum. So when this sperm fertilized with this ovum, this is the genotype of this offspring. So Y besar, Y besar, R besar, R besar. And then you complete all this Punnett Square and you will end up until towards the end. So when we observe the phenotype ratio, the aspect that we could observe, okay, so there are 9 seed with yellow and round. So if you don't trust me, we can count them. So 1 2 3 4 5 6 7 8 9. Yeah, there are 9 seed with yellow color and round shape, and there are 3 seeds with yellow and wrinkle, another 3 with green and round, but there's only one, which is this one, having green seed and wrinkle. So in terms of the phenotype ratio, that's why we have 9 3 3 1. This ratio is very identical, or is what we say, is very common for a dihybrid cross after the F2 generation. Okay. So this is a diagram of representation on the shape and color. Still give you the same phenotype ratio 9 3 3 1. Okay, and in terms of the observation, so there are 2 phenotypes of F2 of this as are the same as both parents. Okay, remember that both of the parents are yellow and round, and the other original parent is green and wrinkle. So hanya ada 2 sahaja yang sama dengan parents dia. The other phenotypes had combined characteristics. Okay, remember there are 4 types of characteristics. Remember 9 3 3 1. Of this 9 3 3 1, so 2 are from the exactly as the parents, and the other 2 are combination of the parents. Okay, so what I mean by combination of the parents, so ada 2 yang sama macam parent dia. Maknanya ada yang 9 9 3 3 1. So 9, the first 9 is having yellow and round, and the last one is green and wrinkle 1. So these 2 are the same as their parents, whereas these 2 are not the same as their parent. Maknanya yellow and wrinkle, mana ada parent dia yellow and wrinkle? Green and round, mana ada parent daripada parent yang asal tu adalah green and round? Tak ada kan? Okay, so for each character, phenotype ratio is 3 to 1. The ratio of yellow seeds to green is also 3 to 1. The ratio of round seeds to wrinkle seeds is 3 to 1. Maknanya ada 3-1. But however, for both character, the phenotype ratio is 9-3-3-1. Okay. So the conclusion for Mendel's experiment is that principle of dominance. Yellow seed allele is dominant over green allele, and round seed is dominant over wrinkle seed allele. And the principle of independent assortment. Each member of a pair allele may combine randomly secara rawak with either of another pair during the formation of gametes, as I've shown earlier during in the diagram. Okay. Now we look at the dihybrid test cross. Ya, dihybrid pun ada test cross juga. Okay, so the genotype capital R, capital Y, capital R, capital Y with small y, and we have capital R, small r, capital Y with capital Y, and also capital R, small r, capital Y with small y, all have the phenotype brown and yellow. These four genotype having the phenotype brown and yellow. Okay, a test cross can be carried out to determine the genotype of the plant. Okay, since because semua 9 ni adalah round and yellow, kita tak tahu yang macam mana dia punya genotype dia. So we need to do a test cross. Alright, if the double heterozygote is crossed with a double homozygous, mean double trait, okay, homozygous for both trait, okay, double homozygous recessive, a genotype and phenotype ratio is 1, 1, 1, 1 is obtained. Okay, so we look on the next diagram. Okay, on the next slide, show you better diagram. Okay, this is what I meant here. So for the F1 test cross, so we have the F1 generation, and we do the test cross. Remember, when we do a test cross, we must cross with homozygous recessive. Okay, and from this is the gametes. Okay, for the F2 generation, i.e. the results after the test cross, it must give you the ratio of 1, 1, 1, 1. Okay, so this diagram showing you the color and the shape of the seed. Okay, still will give you 1, 1, 1 to 1 ratio, same one. Okay. So this is a summary of dihybrid crossing for F1 self-cross. The F2 phenotype ratio is 9, 3, 3, 1. And for the F1 test cross, it will always give you the phenotype ratio for the genotype ratio 1, 1, 1, 1. And same for the phenotype ratio 1, 1, 1, 1. Okay. Alright, so here are the list of the references. Alright, that will be all for this video. Okay, so do drop a like, subscribe, and share this video, and wait for the next video. Okay, thank you.